Hi kids, and welcome to a two-part video on, obviously, DNA technology. Technology is the use of knowledge for benefit, and in this case the benefit for humans. When we take what we learn from science and apply it, we call it technology. We've learned an awful lot about DNA and how it works, and how we can manipulate it to cure diseases and solve all sorts of problems ranging from shortages in food to nutrition. In this first video lecture, we're going to talk about some of that technology, the benefits and the concerns surrounding it. Here are some of our objectives. By the end of this unit, you should be able to define genetic engineering, explain how restriction enzymes can be used to make recombinant DNA, explain how cloning vectors can be used to clone and transfer genes, and list the steps in a gene transfer experiment. Genetic engineering may seem like a new term, but actually humans have been doing it for hundreds and hundreds of years. We've actually selected individual organisms that had traits that we actually desired as humans, allowed those individuals to breed and have offspring like them. When humans do this, we call it selective breeding. It's also known as artificial selection. In natural selection, the environment and nature selects those traits in an organism that make it more suited to the environment. But in artificial or selective breeding, humans are actually choosing those traits in order to get the desired results that they want. Most of your barnyard animals are the result of selective or artificial breeding. Let me show you a few examples of what I'm talking about. A long time ago, humans found a particular plant in nature that produced a product that they saw as a food source. Here is teosinte, grew in North America. Now the natives who discovered this also knew that if they planted the seeds of this plant, that they could get another crop next year. Variations in the teosinte allowed those individuals to select individual plants and seeds that would produce better and better product. And today we know that teosinte is the original corn plant. You may also know that we have lots of different varieties of corn. Here we have strawberries. Perhaps if you've been camping, you've seen wild strawberries growing around on the edge of woods or trails. Strawberries were also selectively bred to produce bigger and bigger strawberries. Here are some wild type tomatoes. This is how they actually exist in nature. The same is true. Humans, over many generations of tomato plants, selected particular traits in the parent plants to produce bigger and better tasting tomatoes. We've done this with all sorts of animals as well. You might know that today's dog was actually bred by humans from the wolf. It's not too hard to see it in some species, but the incredible variety of dog species can make this seem sort of far-fetched. But with our knowledge of DNA and how it works, we can actually show how that's possible. And that's all just through selective breeding. We're not manipulating the DNA in any way. We're just allowing nature to take over, while we, as humans, select the traits that we want in an organism. What can we do now? Well, we can actually now manipulate the genes when they're outside of an organism so that they can do some strange things while in the organism. And we don't do this just out of whimsy or just because we can. Some of these things actually lead to technologies that make life better for us and for the environment. Here's what we call a transgenic pig. A transgenic organism is an organism that has a gene or genes from another species placed within its own genome. These pigs have a jellyfish gene in them. The particular gene from the jellyfish actually produces a protein that makes them glow in the dark. You might think, well, this is a neat idea if you need to find pigs in the dark. But that's not why they did it. They did it just to see if they actually could get it to work. And this illustrates a point. DNA, whether it comes from a jellyfish or a pig or a bacteria, is the same in each organism. And the cellular machinery that allows those genes to be expressed is the same in every single organism. And this is what allows us to do what we call recombinant DNA. That is, putting genetic information from one type of plant or animal into another. This is what creates a transgenic organism. You can also do cloning. Cloning is making genetic copies of existing plants or animals, or even just genes. Let's look at the later of two of these, that is cloning, and how nature has actually taught us how it can be done. All of you are familiar with viruses, but you may not know how they actually work. What a virus actually is merely a membrane or a cell coat, typically made out of a protein, with DNA or RNA on the inside. A virus infects a host cell by merging its DNA with the host's DNA. The host cell recognizes the virus DNA as its own. And as the host DNA cell machinery starts to read that DNA code, the virus actually has tricked it into making more virus. This is what typically makes a cell sick. So when more virus is made by that host cell, it often kills that host cell while the viruses escape to infect other cells. In a sense, the virus has cloned itself inside of a host cell. The fact that viruses need a host cell in order to make more of themselves and can't reproduce on their own is why we don't consider viruses a living organism, but merely an infectious one. Now, an organism is called transgenic if it has genetic information added to it from a different type of organism. It's also known as a genetically modified organism. The acronym GMO is heard more often today. Now with transgenic plants, the work is furthest along. It's easier and less controversial to work with transferring genes from one organism into a plant. We actually have corn now that produces its own insecticide. One of the main pests to farmers who grow corn is the corn borer worm that eats and destroys lots of the crop. It was found that a particular bacteria produced a protein that is actually toxic to this worm. So they took that gene from the bacterium and transferred it into the corn plant. So now the corn plant makes that protein that is only toxic to the worm. And it's increased the yields of corn by farmers tremendously. We have soybeans and cottons that are resistant to herbicides. Now an herbicide is a chemical that actually kills plants. And we spray it on things like weeds. If you can make a crop plant resistant to an herbicide, it's a lot easier to destroy the weeds in a field that are competing with the crop plant if you can just spray them with the herbicide. The crop plant survives, but the weeds die off. Again, increasing the yields of the crop plant. Since the 1950s, papaya plantations in the tropics, especially Hawaii, have been plagued with viruses that destroy the trees that they grow on. They've been successful at inserting a gene into the papayas that make it resistant to that virus. Transgenic crops are being grown in the Americas, South Africa, Europe, Australia, and China. So all over the world. We also have transgenic organisms. Actually manipulate the genes of this chicken so that it doesn't grow feathers. And that has obvious advantages in the meat chicken growing business. But the work is less advanced here. For a long time, human genes have actually been inserted into things like bacteria. The human insulin gene, that is that gene that makes insulin and allows all of us to regulate how much sugar is in our blood, is absent in people who have diabetes. They've been able to take that human insulin gene, insert it into a particular type of bacteria, and get the bacteria to make insulin. The insulin can be extracted from the bacteria, purified, and then used to treat diabetics. Human genes have actually been inserted into mice and other organisms to produce various human proteins for treating diseases. One of the first successes with transgenic organisms is the production of what's called biosteel. It's not really steel, but it is a product made by a living thing. Goats had been given the gene for making spider silk. They wouldn't spin spider silk like Spider-Man, but they would produce the protein in their milk. Now you should know that spider silk is amazingly strong. Those goats, those transgenic goats with the spider silk gene, produced the spider silk in their milk. That protein could be extracted from the milk to make materials like bulletproof vests. Now the advantages of transgenic engineering, that is, creating organisms that have genes in them from other organisms, might seem obvious, but let me point out some of them. We can make plants that are more resistant to disease. We can make plants that have larger yields, that is, produce more product that's more transportable and more nutritious. In fact, we can create a type of rice that has many more vitamins than the old type of rice that we used to produce. And that's important in countries where people are starving and are too undernourished. We can make animals that have proteins for medicinal purposes. Animals that can make organs for transplant into humans if those organs have the correct proteins on their surface so that our immune system will actually not attack and destroy those transplanted organs. But there comes some concern and some dangers that we should think about when we manipulate nature in this way. Here are some of the concerns of transgenic engineering. We wonder if they're safe to eat. Could somehow manipulating the genes of these organisms have an effect on our own genes and cause diseases in us or undesired results? Will they harm other wildlife other than just humans? There is some concern here that the genetically modified plant, when put back in nature, could disrupt the balance in an ecosystem by competing with other plants. Will some of these become super pests or weeds? Some of you might know that the killer bee, while not a transgenic organism, was actually bred by humans in the desire to produce honeybees that produced more honey. That experiment didn't work, and those honeybees, those quote-unquote killer bees, have now entered the ecosystem and are far more aggressive than the wild-type bee in nature and outcompete them for territory and food. There's the idea that some transgenic plants could contaminate natural plants. In animals, well, will those animals be harmful? Will they replace or contaminate natural animals? You can certainly find lots of articles nowadays on the concerns of genetically modified organisms. I encourage you to look it up and to educate yourself on some of the concerns of genetically modified organisms. In fact, you probably find more information on the concerns more than the benefits. I hope that through this class you can actually get a balanced view so that you can weigh the benefits and drawbacks of transgenic engineering. I'm going to end the video right there so that we can take some time to ask some questions in class, have some discussions about the benefits and drawbacks of transgenic engineering or genetically modified organisms. We'll do some activities in class that allow us to see how this is actually done in a laboratory. So if you have any questions, write them down and bring them to class.
Who doesn't know what I'm talking about?